Okay, thank you, Frank. So I'm going to try to be very brief. I'm gonna have only 3 slides and just summarizing what's been happening within the feed additive space, and then to talk a little bit about sort of the delivery mechanisms of feed additives, and then just a little bit of an update of some of the work that we're doing. Just Spencer just mentioned on this audacious project. So we have some results on that. So I will share that with you. This will be the first time that I'm actually sharing these results. So every time I update this, the fonts get smaller and smaller because there's a lot of stuff that's been happening. So the idea is not to just list what's happening there, but to sort of quantify it, give some quantification of those feed additives in terms of their efficacy. So, for example, for the additives like the Bovaer or 3NOP, there's been like 60 or more, or maybe 80 trials that have been done all over the world, and we've done some meta-analysis of that. For dairy, it's already been published in 2023, so it was very well described in terms of the efficacy, depending on the fiber concentration and the dose of 3NOP. And for beef cattle, we just finished it, and we are hoping that it will be at least submitted by end of June. So Karen is part of that team, so we're hoping to get it out of there, and probably a little bit earlier with the preprint service as well. And then with the bromoform-based additives, so that's also seaweed and the synthetic bromoform that was also mentioned earlier, we've seen the efficacy going all the way up to 95%. So the last project we did with ruminate, we saw the oil-based additive going up to 95%. And for this also, now there are about 17 or so papers that have been published in this space. So we put together a meta-analysis on this as well. We just finished it and submitted it over the weekend. I was hoping to share with you the preprints, but the preprint service has not done. It's supposed to be very quick, but it's still taking some time. I'm hoping maybe today or tomorrow you will have a copy of the meta-analysis for bromoform-based feed additives. The tannins, the same thing. There has been quite a number of study that was done. Luis Tedesquez has done meta-analysis on the tannins as well, about 10 to 20% of reduction. We also have updated that meta-analysis with the work that we're doing for USDA. Fernanda is here, Cleves is here, Sarah Place is here. So they are all part of the team where we're trying to figure out what we can recommend to farmers in the US. So that work is also done. Hopefully we will submit it by the end of this week, and then we'll get it up on the web with a preprint service as well. The next one is lipids. For lipids, there's a lot of work. In fact, we found about over 10 meta-analysis done on lipids. So instead of doing another meta-analysis, what we thought we'd do is we do a second-order meta-analysis. So you have a lot of this different meta-analysis, first-order meta-analysis. So we've done a second-order meta-analysis to really dig into what is the level of reduction you can get when you add lipids. So if the diets have different lipids within themselves. So if you start with the 2%, how much further can you go? With the 3%, with the 4%, what's the reduction that you will expect? So that is also finalized. So hopefully we'll submit that by end of June. So we'll get that up on the web soon as well. And then we have others that are coming up in terms of the probiotics, vaccines. I think we have a whole session on vaccines, so I don't need to say anything more. There are other work that's going on in alternative electron receptors, and there are some also algae solutions with algae that do not contain bromoform as well. So lots of work that's been done, and there's more of that I did not include in this slide as well. In terms of how are they being delivered into the animal, a lot of it right now is with the TMR. So that's why there's a lot of focus on dairy. You do it through TMR. There are some slow-release capsules that have been developed right now. I had the pleasure of talking with the New Zealand team yesterday, and they were telling me that there were some capsules that have been almost ready now that they will be able to include. They basically are solution agnostic, so you can put any kind of additive, and then they will be released at at a standard rate. So that would be really great for grazing animals. Leak blocks are also being done, but it's kind of challenges in there. Ruminate have developed a water-soluble additive that I'm looking forward to seeing how it's working well. And then microbial engineering that Spencer just described as well. So, and to my last slide, again, this is the first time I'm sharing. So what we did in this experiment, which is continuing, is that we have this 3 treatments, 1 control. One is we give the animals seaweed, particularly brominata, until they are weaned, like maybe 1 or 2 weeks after they are weaned, and then we stop the additive. And the third one is we start the additive after they are weaned. So you can see the control up there, and then the ones that we gave the seaweed and we stopped, about 4 months after we stopped, they were still reduced. They still had 30% reduction in methane emissions. It was starting to come up in the 5th month, and then it's going down in the 6th month. So I'm not sure what's going on there, but the number of animals here is quite small. So we're going to be adding more and more animals. We have 24 animals in total. So what you see here is probably 5 or 6 animals. So as we have more and more animals into this data, and then we'll continue this until slaughter. So this is going to be 18 months study. We are about a year into this study right now. So next year, this time, if I get invited, then I can I can share the whole thing. Hopefully, by that time, we will almost be finishing. So this is really interesting in terms of early intervention, and that was mentioned earlier as well. When you do early intervention, yeah, you can really reprogram the rumen. Thank you.